Okay, so we are going to be starting now, and today we're going to be learning about the immune system. I don't know, I just found out something cool about my background. I can change it. Yes, as you can tell, we're going to be learning about the immune system. So this is honestly a very interesting topic that covers really how your body defends itself, because we constantly get a lot of different viruses and bacteria and things like that, and it's really what your immune system that helps protect you from that, right? And our immune system's actually like very overly complex, especially since it isn't just like COVID-19 nowadays or viruses nowadays. Back then, especially in the older days where we didn't have such efficient treatment, viruses and bacteria were such a large problem. And that's what led to our immune system to be very complex and very well thought out and very well evolved, you could say, in a way that helps prevent these viruses from affecting you to the best of its ability. Okay, so the immune system. So what even is your immune system? Well, your immune system is the body's natural defense against illness. It protects you from harmful invaders like bacteria, viruses, and other germs using a network of different cells, tissues, organs. It includes things like your white blood cells, which we talked about in our circulatory unit, and the class where we talked about the different components of the blood, and we touched on white blood cells, and this is what really helps you fight the infection. It involves things like antibodies. I'm sure you guys heard of what antibodies are, and we're going to be explaining how that works and what that is today. Many other tools that work together to find and destroy these threats. Even simple things like proteins, helping you stay healthy and fight off those infections. So just a brief overview of what the immune system is mostly split into. So the immune system is split into two main sections. First, we have the innate immune system, and then we have the adaptive immune system. So the differences is the innate immune system is not specific, and we'll explain what that term really means later on. But the innate immune system, basically, it's like your first line of defense, right? It doesn't matter what virus it is, what bacteria it is, and it doesn't really specifically target it. It just eats any foreign invaders. Like, for example, this includes things like your skin, things like your fever response, things like actually you have like cells as well that are part of the innate immune system, which is like your macrophages and your neutrophils. But you also have cells in your adaptive immune system. So your adaptive immune system is more specific and it's a bit more complex, and we'll explain why later. But essentially, it uses what things called antibodies, and these antibodies are very specific. We have millions of different types of antibodies that are on B cells, which is a type of cell, and these antibodies, there are many, many different types, and they recognize a specific antigen, which is basically something on a virus. And once they find the specific antigen on the virus that matches with the specific antibody, these antibodies can help fight the infection against the virus better. So the adaptive immune system is the really powerful one that helps really defeat the virus, but it takes time, and that's why we have the innate immune system to take care of it first and to kind of lessen the load. And also, the adaptive immune system cannot function without the innate immune system, and we'll explore why that is later. It has to do something with like presenting antigens and etc. Um, and the adaptive immune system is also further split into two more sections, and we'll talk about that later. So your first line of defense is your skin. So your skin blocks germs, kind of like your natural shield. Um, you notice that you sweat, and then you have your sweat glands as well, like a sweat pore. And sweat is actually contains oil, and it's somewhat acidic, and that's why it can kill some bacteria. Then you have things called Langerhans cells, and it's just kind of immune scouts in your skin. They're basically kind of like macrophages, except they're more specialized in detecting invaders on your cells. So it's kind of like outside guard cells that don't really attack the germs, but they help kind of scavenge for them. Also, your skin, there's this place called the lower epidermis. I'm not sure. Yeah, this picture doesn't show it. But somewhere down here, your skin is constantly like uh it's like dead skin is coming off the top consistently, and then new skin is being formed by your dividing cells via mitosis, like we explained before, on the bottom. So that's why, like, if you were to scratch your skin, a lot of skin is actually coming off with that, and that's just dead skin. The surface of the skin is all dead. Um, and you might be wondering, then why do I feel pain? Well, the cells below your skin are actually alive, so that's what allows you to feel pain. That's through like receptors, like we talked about in our neurology class. And so skin is constantly being shed off. So if you can think about it, although you can't really tell that it's shedding off, it's pretty slow. But if like bacteria, viruses, and germs were to be on your hand or say your arm, the skin is constantly shedding itself. So therefore, the bacteria and the viruses are more likely to fall off. It also sends alerts when cut. So this is what is leads to inflammation, and that's like the swollen. That's why usually if you get like a cut, like a paper cut, probably not paper, but something like a bigger cut, the skin around it will start to swell and it'll start to become warm, right? And that's what we call inflammation. And what inflammation does is it allows more blood and more water to rush into that area. And remember, blood contains your white blood cells, so that allows the immune system to fight back better to the cut spot. Because the cut spot is where germs and bacteria can get in, right? Because it's breached the security system of the skin. It also has tiny germ-killing proteins that it makes. These are what we call part of the complementary system, which we'll explore later. And this is just a group of proteins, individual proteins that help fight these bacteria. The skin also can seal wounds, like if you get a cut, it obviously heals itself. And it also defends while healing. So if there's ever a scratch and you're healing, the body puts even more specific emphasis onto that area. Just a quick shout out to what we call mucus. Sorry for the lag there. I'm like, the Wi-Fi is kind of bad over here, but shout out to the mucus. And so the mucus is what lines most of your donut hole, you could say. So I like to think of humans as basically a giant donut, like we explored last class, from the mouth to the expel, the anus. That's basically just a giant tube, and so all in all, we're just a big donut, right? And so the mucus is what lines this donut. So it lines like your nasal cavity, your small intestines, right? And mucus just helps trap germs and viruses. And they're packed with things like enzymes, antibodies, and even immune cells. And so you could really see that once a virus gets stuck to the mucus, it has a bunch of ways to destroy it within. And it blocks microbes from reaching deeper tissues. So if it's just in like your small intestine, it won't cause that big of a problem. But if it gets leaked through your intestinal walls, for example, then that's a problem, right? If it gets into the bloodstream, that's an even bigger problem. So this mucus helps prevent that. Like I said, it's found like things like your nose, throat, lungs, gut, and it just covers everything, total coverage. It also covers a bunch of your lungs, your tracheal spots, because obviously that is important as well. That's another opening. It also works with something called cilia, which we talked about, which are those like little hair-like projections. And that helps really sweep out the gunk. So this is very it can be very seen in like your throat, etc., where the thing can get swept out. And also mucuses in general keeps surfaces moist, and we know that everything likes to have water near it to help keep surfaces moist to protect and heal just in general. Next, we'll talk about inflammation. So this is kind of your secondary response, assuming the skin gets bleached. So let's say you're cutting and you cut your finger just a bit, right? It looks kind of deep, but it's not that deep. Um, let's just assume you cut it a little bit with a knife while you were chopping, say, cucumbers. And so what essentially what happens is that let's say the knife, because you were chopping something else before. You were chopping pork belly, right? And say that pig had some sort of bacteria on the knife. And so the knife would come into your skin, and then it would kind of give your body the bacteria. And what directly happens, not really related to the bacteria entering your body, but what directly happens is that this knife comes in contact with things called your mast cells, M-A-S-T, mast cells. And these cells essentially will send out something called chemokines. And these, like we talked about before, are those little signaling molecules that lead to vasodilation, right? And so as you can see here, these molecules lead to vasodilation, which essentially just means it's the widening of the red blood cells of the blood vessel. And so that allows more red blood cells, more fluid, and most importantly, more white blood cells to flow into that infected area so that it can fight off the bacteria more effectively. Also, we talked about this before, not directly related to the immunitary response, but we also have platelets leaking in. So that's another important factor of this inflammation process, right? It allows platelets to also help seal the plug. Next, I have a small video talking about fever, and this is your kind of third line of defense. And why do you even get a fever? What's the point? Why does your body want you to get a fever? I'm gonna quickly unshare and share again because I want to make sure I'm sharing with sound. Here.
Hi, this is Kate from Minute Earth. When you get an infection, your body will often get to work generating extra heat, which we know as a fever, to help you fight the pathogen, which seems logical. After all, we also heat raw food, questionable water, and used surgical equipment to literally sizzle pathogens to death. But that's not how a fever works. I mean, even the puniest strategies for killing germs with heat require temperatures of around 65 degrees Celsius, yet most fevers top out at 40 degrees or so. Any temperature that's hot enough to decimate an invasion of viruses or bacteria would also decimate your own cells. A few extra degrees can slow down the replication of certain pathogens, but the bigger effect of a fever is how it benefits our own bodies. When your body temperature rises, neutrophils, white blood cells that lurk in your bone marrow, wake up and start gobbling germs. And other germ gobblers get better at identifying and engulfing pathogens when your temp is higher. Then there are T cells, which travel to lymph nodes to identify the pathogen, but they have to get there quickly before the germ overwhelms your body. When your body heats up, T cells get there way faster and can coordinate an effective response in time. But why does heat, and not even all that much heat, have all these effects? The answer, paradoxically, may have to do with the dangers of too much heat. If you're, say, jogging in the summer and you start to overheat, delicate components inside you can start breaking down. So as your temperature rises, your body jumps into action to protect itself. For instance, cells churn out tons of heat shock proteins, which find molecules that are unraveling and bind to them, either to stabilize or re-ravel them. When you get an infection, a fever may work, at least in part, by actively increasing your body temperature in order to trigger the same kind of protective process. Your body churns out those same heat shock proteins, which are so good at finding and binding to all sorts of molecules. They bind to molecules on the surface of T cells, creating sticky complexes that help the cells grab onto blood vessel walls and make the trek to the lymph nodes, where they ID the invader and coordinate a response. Heat shock proteins can also recognize and bind to specific bits of pathogens, sometimes to the heat shock proteins the pathogens themselves have churned out, then transport them to waiting germ gobblers. Our ancestors first co-opted these existing protective mechanisms to help fend off infections as long as 600 million years ago, and animals have been turning up the heat on pathogens ever since. Even critters that can't regulate their body temperature internally will bask, buoy, or buzz until they're toasty and infection-free. And no matter what species you are, the strategy works. Infected individuals that boost their temps recover more quickly and more often than those that don't. So the next time you get a fever, make sure you give it a warm welcome.
Okay, so hopefully you enjoyed that video. Next we'll talk about your fourth line of defense for the innate system, kind of, and that's your macrophages and your neutrophils. So, these can get a bit confusing on the difference between the two, but essentially macrophages are kind of larger and they're more sophisticated than neutrophils. So these are actually cells that are part of the innate immune system, and here's what they do. So first we have the macrophage. This is what macrophage would kind of look like under a microscope after staining. And so the macrophage, oh, I think these dot dots are off again, but essentially I'm just gonna explain it quickly. The macrophage kind of uses its like tentacles, you could say, and it kind of moves around a bacteria. I'll show you a GIF of that later. But essentially it consumes these bacteria and these viruses, it kind of engulfs them. At the inside this body, it destroys them. So it's kind of like it's literally just like a big eater that kind of eats all these bacteria up and these viruses up and it destroys them. So how it does it specifically is here we have a virus, and as you can see, it extends its body via amboid motion, AMEOB, something like that. That type of motion you can search it up, and essentially it uses the cellular exoskeleton, which are those like little fibers, like your microtubules, your microfilaments inside the cell, and it helps extend the cell membrane so it can grab onto one of these viruses, kind of eats the virus in, and then this kind of membrane forms, like as you can see, a little like vesicle. We literally just call it a vesicle inside the cell. And then what happens is you also have your lysosomes. We talked about this in our one of our first classes. And these lysosomes, remember, contain those digestive systems, and they're also more acidic. I think if you guys remember, I showed you that little like graph of which parts of the cell are more acidic, and the lysosomes were one of the most acidic parts, and because it is used for digestion, and more specifically in macrophages for the destruction of the bacteria or the virus. So you can see after it enters, these vesicles kind of combine, very interesting. And these lysosome enzymes and the acidic environment helps destroy the bacteria and that's expelled. And what's really interesting is the body can actually use this material because remember, everything is made up of around the same things, right? DNA bacteria have it, RNA bacteria have it, and all made of nucleic acids. Membranes, they have it. So kind of the body actually could like kind of eat these viruses and then spit them out in smaller molecules that are no longer working as a virus, obviously. And then these smaller molecules can then actually be used to build some of the body cells and help them against the infection. So this is just a quick GIF. You can see this macrophage is chasing this virus. I'm not sure if it's a virus or a bacteria. It looks like a bacteria. So it's chasing the bacteria here. And as you can see, it eats it. So this is just a quick, cool GIF to show you how these macrophages like go about eating these bacteria. And this is obviously sped up a lot as well. Next, we have neutrophils. So these are your macrophages are actually not the first responders to infection, the cell responders. Neutrophils are actually the first responders to infection, and they specifically target bacteria, not so much viruses, and they kill pathogens by using enzyme and reactive oxygen species. And so that's just a fancy term for a different type of neutrophil. So essentially, they still engulf these bacteria, but they have to self-destruct in order to kill them. So they're a bit less efficient and less complicated than macrophages, but they are much more plentiful. Like they engulf microbes, yes, they also release something called the NETs, which are neutrophil extracellular traps. We'll explore that very briefly. It's pretty complex. It's very interesting. It's kind of like the neutrophils shooting out like a web kind of, and it just helps trap pathogens. Pathogens is just so you guys know, the term pathogen is just a general term for anything that affects your body negatively, like viruses, bacteria, fungi, whatever. And so yeah, these extracellular traps helps trap those pathogens, so then the neutrophils and macrophages can eat them better and easier. They're very short-lived and mostly are only mass-produced after infection. So after the skin cells kind of know that something's wrong, it gets mass-produced. Yeah, like we talked about, it's the most abundant white blood cell in circulation, probably because it needs to be abundant because it kind of self-destructs upon ingesting a couple bacteria. Another very kind of off topic, another very interesting thing we can see from this graph is if you look to the side, these are red blood cells. So this is a sample of blood, and you can see how this person might have symptoms of actually anemia, which remember is problems with the red blood cells. As you can see here, the inside is very hollow for some of these red blood cells. So this may indicate something maybe related to cancer, but I think this person might actually have anemia, and that's why the red blood cells kind of look have this very bland center as well. Kind of off topic, but just a key thing to think about, right? How this all connects to maybe a more real world application. And like again, for example, if you took a blood sample of someone, right, and there were a lot more neutrophils than normal, that can also indicate an infection. This is just like a kind of cool animation, cool like not really animation, but a cool picture I found online, just about a neutrophil and just how it eats bacteria essentially. So this is the neutrophil extracellular trap. So it essentially just kind of breaks a part of its enzyme and it just releases a bunch of its fluid, like platelets, myeloperoxidase, which is just a fancy term for something similar to a lysosome that helps break down bacteria. You have your neutrophil elastase, which also helps kill bacteria. You have your fibrin, like we talked about. Platelets and fibrin work together to form a clot, and these neutrophils also take this to advantage to form a kind of sheet that helps keep everything together. It also releases histones because it releases some of its DNA strands as well, helps stabilize everything. Next, we have our natural killer cells. These are also part of the innate immune system. These are all part of the innate immune system. We still have not moved on to the specific adaptive immune system. So these are, you could tell that the neutrophils and the macrophages were specifically targeted to killing things outside of cells, like in your blood, in your tissues, right? But these natural killer cells they kill viruses and bacteria that have already infected your cells. So I'll tell you how viruses affect your cells briefly near the end, but all viruses need to infect your cells, right? Obviously, they need your cells to reproduce. Not so much bacteria. Bacteria can produce by themselves through things like binary fission, but viruses really need the host cell to reproduce. And so these natural killer cells are there to help kill these cells and signal them to self-destruct through apoptosis by releasing toxic granules into them if they contain a virus. So for example, let's just say this cell contained a virus. The T cell would use these receptors called the KIR receptor to kind of bind to the cell and kill it. This kind of complimation here, this is just like to recognize that it's actually a cell and not anything else. And this helps bind the natural killer cell to the cell, and so that this other cell called the KIR receptor can match with the MHC1. So the MHC1 molecule is very, very interesting. This molecule essentially takes fragments of foreign stuff and it puts it on there. It's like if a virus went into the cell, the cell would like take one of its components and put on an MHC1. So if this MHC1 with the virus part it is attached to it, that means it's a healthy cell. So because the natural killer cell has these receptors and they're built for detecting healthy cells. The healthy cells will display a normal MHC1, but an unhealthy cell will display a MHC1. This diagram is a bit weird. It doesn't really show another MHC1 molecule, but this in effect an infected cell would show an MHC1 molecule, but this green bit here, let's just say, is red, right? It's different. It shows the virus's MHC1. That's not good, right? This won't bind, and then it will release these toxic granules that would allow the cell to die. Here's another quick video about oncogenes. And oncogenes is basically the study of cancer. And so this T cell and everything, it really connects to how cancer works. And I thought it'd just be a really good idea to include a video on what cancer is. It's about 11-12 minutes. I think it's very interesting. It really explains what cancer is. And it also really helps you to think about what cancer is. Because like we've obviously heard a bunch of people unfortunately pass away due to cancer, how cancer research is such a big thing, how we haven't found a cure for cancer. And I really want you guys to actually understand what is cancer, and so you guys can have a better understanding of like just general life knowledge. And it's just cool to understand how something so deadly like even works and what it is.
Hello, in this video, we're going to talk about oncogenetics, the mechanism of cancer from a gene point of view. In order to understand this, we have to learn again about the cell cycle. So a normal cell can just be at rest. This is at a quiescent phase, the G0 phase. The cell can then enter the cell cycle. The first phase of the cell cycle is known as the G1 phase, the growth phase. This is where the cell's organelle duplicates. So here you can see the mitochondria of the cell is duplicated. After the growth 1 phase is the S phase, also known as the synthesis phase. This is when the DNA duplicates. After the S phase, there is a G2 phase, where the cell essentially grows again, the growth phase, and prepares itself for the M phase. The M phase is also known as mitosis, where a cell, which is now ready, essentially divides into two identical daughter cells. These new cells can then re-enter the cell cycle or go back to the G0 phase. Now, the cell cycle is, as it looks, a continuous cycle. However, things can go wrong throughout the cell cycle, and so it's important to have checkpoints to make sure that there are no problems along the way. The first checkpoint is actually at the end of the G1 phase, called the G1 checkpoint. This is to make sure that there's no problems in the DNA and in the cell itself. The second checkpoint is at the G2 checkpoint. This is to make sure that the cell has no problems before it enters mitosis. And then there's another checkpoint at the M phase as well. The cell cycle is a continuous progression from G1, S, G2, and M. But what actually drives the cell through the cell cycle? Well, when a cell enters the cell cycle, it will start making proteins, allowing it to go and progress through the cell cycle. These proteins are your cyclins and your CDKs, which are the drivers of the cell cycle. So, for example, a cell wants to enter the cell cycle. The cell will start producing proteins, the CDK and cyclins. In the early G1 phase, CDK 4 and 6 are produced, and when cyclin D binds onto this, it will cause a reaction to occur inside that cell. It will cause E2F to detach from the retinoblastoma protein. When E2F is released, it acts like a transcription factor, allowing that particular cell to progress through to the S phase. However, at the end of the G1 phase, before the S phase, there's also another CDK and cyclin, CDK2 and cyclin E. Once at the S phase, the cell will produce another CDK and cyclin, CDK1 and 2, and even cyclin A. And then at the G2 phase, again, CDK1 and cyclin B. And these CDK and cyclins, again, will allow the cell to progress through the cell cycle. So, CDK and cyclins are the drivers of the cell cycle. If you have too low amounts of CDK and cyclin, the cell doesn't really progress through the cell cycle. But if you have too much cyclin and CDK, then you get these cells that continuously enter the cell cycle, and thus you get this uncontrolled growth of cells. And this is one of the mechanisms of cancer. So what can potentially cause an increase in CDK and cyclins within the cell? So this is where genetic mutations come in. So the mechanism of cancer, genetic mutations. So let's just look at this normal cell and pull out its genetic material, which is DNA. DNA is a double-stranded helix made up of 4 types of nucleotides. Now, mutations can occur within the DNA, which will cause changes to that cell. Some types of mutations include point mutations, a single change in a nucleotide. Another is what's called DNA amplification. When a certain gene gets amplified so many times, it could be a bad gene, for example, Then there's another one called chromosomal rearrangement, where the chromosome basically attached to one another where it shouldn't. And another one is called epigenetic modifications, such as methylation and acetylation above genes. And this can essentially silence certain genes and even cause genes to become more active. And so you can imagine with these mutations, a normal cell can become a cancerous cell. And so when the cell enters the cell cycle, you get an uncontrolled cell growth. It bypasses all the checkpoints, you get uncontrolled cell growth. And these uncontrolled cell growth is essentially caused by two main changes that occurs in cancer cell. These are, one, activation of oncogenes, such as the RAS gene and your MYC gene. The other change is the inactivation of tumor suppressor genes, such as inactivation of p53, APC, and BRCA1 and 2. So the mechanism of uncontrolled cell growth, as we discussed, the point mutations, the gene amplification, the chromosomal rearrangements, the epigenetic modifications, these mechanisms of uncontrolled cell growth essentially causes two main things in the cancer cell. These are activation of oncogenes and inactivation of tumor suppressor genes. So now let's look at each of these in a bit more detail, beginning by looking at oncogenes first. So let's begin by looking at oncogene activation, looking at the RAS and MYC gene as an example. So let's look at this cell here that's about to enter the cell cycle at the G1 phase. Now, normally our cell contains DNA, and normally our cells contain a gene called the RAS gene. Now, the RAS gene makes the RAS protein, which basically is an intracellular protein that sits below the plasma membrane. Next to it is a receptor, the growth factor receptor. Now, obviously, normally, when a cell enters a cell cycle, there is a growth factor which stimulates the growth factor receptor. When the growth factor is stimulated, it will actually activate the RAS protein. Once the RAS protein is activated, it will cause a cascade of intracellular phosphorylation of other proteins, which will essentially, at the end, activate a transcription factor. Once this transcription factor is activated, it will essentially go to the DNA and read the genes to make proteins for cell growth, particularly to make proteins to allow this cell to go from the G1 phase to the S phase. And these proteins are the CDK and cyclins we talked about. So you can imagine what would happen if you have a mutation in the RAS gene. When you have a mutation in the RAS gene, you are making actually RAS proteins which are already activated. And so you get always this cascade of phosphorylation events, and you always get the activation of these transcription factors. And so you are overproducing at the end these proteins for cell growth, such as the cyclins and CDKs. Now the MYC gene is another one. The MYC gene normally makes proteins in our body. These proteins are important for cell growth, cell survival, and also cell activity. And so when you have a mutation of the MYC gene, the cell becomes more cancerous. You get more cell growth, more cell activity, and more cell survival. And so activation of these oncogenes, activation of the RAS gene and the MYC gene, for example, will allow a cell to bypass the checkpoints of the cell cycle and will allow the cell to have an uncontrolled cell growth. Now, normally the cell has a mechanism to stop any abnormal cells from progressing to the cell cycle. This is where tumor suppressor genes come in. So for example, let's just say the cell gets held up at the G2 phase because it has an abnormal DNA. It has a damaged DNA. This cell that was stopped with the damaged DNA will not progress through the cell cycle because it is abnormal. It has a damaged DNA. So now let's talk about how this happened. Let's talk about tumor suppressor genes normally, focusing on p53. So let's zoom into this cell. The cell contains damaged DNA. When there's damaged DNA, the cell produces p53 proteins, which can act like a transcription factor. it will read the DNA and will actually make proteins. It will make proteins for cell arrest, such as P21. What does P21 do? Well, P21 is a protein that causes cell arrest. It actually inhibits the CDK and cyclins, and thus it inhibits the drivers of the cell cycle, so the cell cycle will not progress. P53 will also make proteins important for cell repair. And so, hopefully, when the cell is arrested, the cell can repair itself. It can repair the DNA. The P53 protein will also make proteins important for apoptosis. If the cell cannot repair itself, it has to die because we don't want any abnormal cells. So you can imagine now, if you have inactivation of tumor suppressor genes, such as inactivation of P53. When you have inactivation of P53, you are not making proteins for cell arrest. You're not making proteins for cell repair. You're not making proteins for apoptosis. And so you have this cell that enters the cell cycle and can bypass the checkpoints and continuously grow and proliferate. And so in summary, the genetic changes that occur in cancer are the inactivation of the tumor suppressor genes and the activation of the oncogenes. And so you have this cell that enters the cell cycle and can bypass the checkpoints and continuously grow and proliferate. I hope this video was helpful. Thank you for watching.
Cool. So hopefully you learned something from that. I know it was a bit long, but I think he really explained well how cancer kind of works. It is a bit complex, but I just really wanted you guys to get the general idea. So next we have what we call complement proteins. And these are kind of just pure proteins that help your body fight off infections. And so they kind of tag pathogens, and it's a process called opsonization. And so this is basically just the process of proteins kind of tagging the bacteria or virus, and this makes them easier for targeting. It also can form things called membrane attack complexes, as you can see here. And this is just, it's so cool to see this immune system at work. It's basically just these proteins that surround kind of the cell membrane and they puncture a hole. And so this creates membrane problems and it helps destroy foreign pathogens. It also helps trigger inflammation to recruit immune cells. And it kind of works in a cascade. So one activates the next. So it's like it really helps amplify the protein. So finally, we get to what we call the adaptive immune system. So before, all we were talking about was the innate immune system. Now we're going to talk about the adaptive immune system. So the adaptive immune system comes in two major parts. First, we have the humoral system, and second, we have the cell-mediated system. And so the humoral system consists of things like your antibody, antigen, your lymphocytes, your B cells, etc. And then your cell-mediated system contains more of like your immune response based off of cellular interaction. So like that's your kind of cells, that's your other cells kind of killing your own cells. And you can see here that I did say natural killer cells were part of the innate immune system, but actually the natural killer cells is kind of in the middle. It's like in between the kind of innate and adaptive immune system. It's kind of weird because technically its antigen is kind of detecting the viruses like outside exterior, so it has to be specific. It's adaptive to the bacteria, but then again it's not really like kind of directly to the bacteria. So this is kind of in the gray area. And so you'll see this kind of theme pop up in like many different topics as well. This kind of gray area where like you can't really know if it's specifically in one section or another. So like for example, this one, you don't know if it's an innate or adaptive, right? So it's kind of it's really weird. And so again, we have another video I think I will be adding a bit more videos into my lessons now just because I think they're a really good way to explain stuff. And this video just talks about the main differences of what the humoral versus cell-mediated immune system is. So I'll just go over some brief overview, and then I'll dive a bit more deep into it after.
This video is immunology series video. In this video, we'll be discussing about humoral immunity and cellular immunity. Particularly, we'll be more towards their differences. To understand what's humoral and cellular immunity, we see when the pathogen enters your body, it has to counter your immune system first. And in that case, the immune system launches the attack for the pathogen to be eliminated. Our immune system either launches attack by making antibodies to counter the pathogen, or the cells itself engage to eliminate the pathogen. And the most important thing is that the antibodies eliminate the extracellular antigens or pathogens that are circulating in your circulatory system, while some immune cells engage to eliminate the intracellular antigens. So immune system works in two ways. The antibody mediated immunity forms the humoral type of immunity, while the immune cell mediated immunity forms the cellular immunity. Now let's see in detail how both mechanisms occur side by side. The most important difference between humoral and cellular immunity is that humoral immunity is antibody-mediated immunity, while the cellular is cell-mediated immunity. The second difference we see is that humoral immunity eliminates extracellular pathogens or antigens, while the cellular immunity eliminates intracellular pathogens or antigens. In humoral immunity, the B cells play a major role, while in cellular immunity, the T cells play a major role. Now let's see the humoral immunity in detail first. In humoral immunity, we see when there is presence of any extracellular antigen in our body, it triggers the immune system to defend against this antigen or pathogen. And the B cells of our immune system comes into action. The B cells binds to the antigen for which it's specific via receptors. After the binding of the B cell requires some help from T cells. So in that case, the help is given by the T helper cells from the cellular immunity. And these T helper cells secrete cytokines, and these cytokines from the T cell transform B cells of humoral immunity into antibody producing cells called plasma cells. And you know the plasma cells have capability to produce antibodies. So plasma cells produces the antibodies for the specific pathogen. After that, the antibodies neutralize the extracellular pathogen and eliminate it. So this is how humoral immunity works. Now getting to the cellular immunity, we see the intracellular pathogens are displayed or flagged by antigen presenting cells via MSC processing. Intracellular antigens are expressed on the surface of antigen presenting cells like macrophages or dendrites. In the first step, the T cells bind to the MSC antigen complexes on the surface of infected cell, activating the T cell with its cytokine receptors. The activated T cells secrete cytokines, which in turn activates T helper cells and macrophages. After that, the T cell transforms into cytotoxic T cell. And this cytotoxic T cell induces apoptosis in the infected cell and eliminates the intracellular pathogen via apoptosis. So in this way, the cell-mediated immune system works. Also remember, some T and B cells differentiate into memory cells that respond rapidly to any secondary encounter with an antigen. So this is all about humoral and cellular immunity. I hope you like the video. If you like it, give it a thumbs up and make sure to subscribe this channel. Thanks.
Yeah, so hopefully you found that video interesting as well. It just kind of briefly outlined like the major differences between the two. Next we have, so next I'll just dive a bit more into what antibodies actually are. So essentially the brief overview of the structure, I don't want to go too deep into this, but we just have a kind of heavy chain over here. We have our variable regions and we have our light chains over here. And this area called the variable region is where the antigen kind of binds. And this is where the other side is. So we can essentially see how the binding sites kind of bind to these viruses. So although this is That's kind of low quality, but essentially what this does is like the only reason viruses can actually enter cells is they have these surface receptors, right? They have these receptors on their surfaces that bind and then they kind of unlock the cell. But what these antibodies allow it to do is they kind of attach to these binders themselves, and so this prevents them from entering cells. Very helpful. Another thing that's very interesting is it helps kind of aggregate viruses. And since viruses and bacteria are very small compared to a macrophage, what antibodies usually do is they help kind of group all these bacteria together into a clump, and this allows the macrophages to better eat them. So this is remember how we talked about T cells and apoptosis? This is the pathway for apoptosis. Yeah, I don't even understand this. It's very, very complex. And the only reason I put it here was to just show you how complex things are in biology and how like there's a lot to learn. And yeah, it's just like a very complex pathway for just a simple process called apoptosis. Many different things are involved, and it's truly amazing how biology has really evolved this way. So I just wanted you guys to appreciate that once again. And remember, apoptosis is the death of the cell. Just some key enzymes that I see here that I just want to highlight that will definitely that you guys heard of in especially in the cancer video that we watched is here we have the RAS gene over here. And remember, like the apoptosis pathway is very important. That's why you see some enzymes here you see in the that you saw in the cancer video, because apoptosis, like you talked about, is very important for the cancer cell, how cells need to undergo apoptosis or else they will go into cancerous cells. And so some notable enzymes we see here are RAS enzyme. Here we also see, let me see, our Cas-P6. This is a very important protein that regulates a lot of things, all of this random stuff. And then we have our P53. You've definitely heard of P53 in that video. It's very, very important. And some other different ones. Cyclin C, also pretty important. And then we have lysosomes, and this is just a bunch of different pathways. And then obviously our surface receptors. And so finally, we'll just talk about viruses. So viruses just come in four major types. We have our bacteriophages, tobacco mosaic viruses, adenoviruses, and influenza viruses. So they all look like this. And viruses, essentially, they bind to the receptors, they go into the cell, and they release their cellular DNA slash RNA. And so essentially, the cell kind of absorbs this DNA slash RNA. And what it does is viruses kind of hijack the cell's machinery. So it takes the cell's ribosomes to use its DNA to clone the DNA, and then use the ribosomes of the cell to take its DNA slash RNA and make it into its other proteins that then assemble, go into the membrane, and then disperse. So you can see it's kind of cool how it like sticks to the surface and then it kind of pops off. Yeah, that's basically the end of my presentation. The final thing that I'm going to show you is just a brief overview of the immune system by Cook's.not. And I love his visuals, obviously. So I think this will really help wrap up our lesson, as we don't have much time left anyway. So we'll just finish this video. We might play a booklet next class because I did make a booklet, but we are essentially out of time. So I'm gonna play this video. Really helps you visualize it much better.
The human immune system is the most complex biological system we know after the human brain, and yet most of us never learn how it works or what it is. Your immune system consists of hundreds of tiny and 2 large organs. It has its own transport network spread throughout your body. Every day, it makes hundreds of billions of fresh cells organized like an army, with soldiers, captains, intelligence officers, heavy weapons, and crazy suicide bombers. It's not some sort of abstract entity. Your immune system is you. Your biology protecting you from the billions of microorganisms that want to consume you, and from your own perverted cells that turn into cancer. It's so manifold that it's impossible to cover in one video, so we'll make a series looking at different aspects of it. Today, what happens when your body is invaded and your first lines of defenses are engaged in a fight for life and death? been a normal day when suddenly the world explodes and an asteroid rips the sky open. Countless alien life forms invade, ready to destroy cities and infrastructure and eat civilians. Or this is what your cells experience. You look at your bleeding thumb that you just cut on a dirty twig in the park. How annoying. But inside the wound, a horrible catastrophe has happened. There are dead cells and blood and dirt everywhere. Even worse, countless bacteria invade the warm caverns between your helpless cells to explore their new home, steal your resources, and poop everywhere. Immediately, the first stage of your defense kicks in. The cells that survived the impact or are hurt or dying scream in panic, releasing an onslaught of chemical alarm signals that awaken your immune system. The first cells to show up are macrophages. If an average cell were the size of a human, a macrophage would be the size of a black rhino. A stoic cell in principle, but you wouldn't want to annoy it. Bacteria do annoy them. Within seconds, the large cells attack and begin killing them without mercy. They stretch out parts like the arms of an octopus and grab the bacteria to swallow them whole and digest them alive. A macrophage can eat 100 bacteria before it's exhausted. But there are too many enemies, so the macrophages call for reinforcements. In your
This video is gonna go over time, so if you need to leave, feel free to leave. I'm gonna stay in the call and finish this video, and then I'm gonna end it there, so you guys can leave if you guys want.
blood, hundreds of thousands of neutrophils pick up their signals and move to the battlefield. Neutrophils are intense suicide warriors that only live to kill. They're so enthusiastic about killing that they kill themselves a few days after birth, so they don't have time to accidentally destroy your body from the inside. As soon as neutrophils arrive, they begin vomiting deadly chemicals at bacteria or devour them. They are so careless in their attacks that they are causing real damage to your own cells, but collateral damage is not their concern now. Or ever. Some neutrophils go so far to push their suicide button and explode, casting wide and toxic nets made from their own DNA filled with deadly chemicals that trap and kill bacteria. Sometimes they can continue fighting after that, even though they're sort of dead already. This is how much fun they have killing. While the battle rages, your blood vessels let fluid stream into the battlefield like a dam opening up towards a valley. You notice this as inflammation. Your thumb swells up a little and gets red and warm. The fluid brings a silent killer into the battle zone, millions of complement proteins, a sort of automated liquid weapon that stuns and kills bacteria by ripping holes into them. We made a whole video explaining them in detail. We're reaching a crossroad now. If things go well, your first line of defense kills the invaders quickly. But sometimes the enemies are too strong and would overwhelm your defenses eventually, which means certain death for you, the human. This is the hour of the dendritic cell, your immune system's intelligence officer. While your soldiers were bashing in heads, it was collecting samples by ripping bacteria into tiny parts and covering itself in it. Like a soldier decorating itself in the guts of a dead enemy. The cell leaves the battlefield and enters the superhighway of your immune system that connects all your tissues with your immune headquarters, your lymph nodes. The dendritic cell coming from the battlefield is looking for a helper T cell, which is a sort of all-purpose commander cell within your immune army. But not any helper T cell, one that happens to have just the right weapon for the bacteria that infected your wound. So it goes around and rubs itself, still covered in bacteria parts, against every helper T cell it meets. Most T cells are a bit disgusted and not interested, but after a few hours, something clicks. A helper T cell recognizes the bacteria parts. This cell is the weapon that's needed right now. The dendritic cell is overjoyed and activates the helper T cell. Okay, wait. How come your immune system has a cell that has a weapon against the specific bacteria that infected you? Well, your immune system has a perfect weapon against every possible disease in the universe, against the Black Death, the coronavirus, or an infection that will emerge in 100 years on Mars. We'll talk about this a bit more in the next video because it's very complex. So for now, just know that you have billions of unique helper T cells that each have weapons against every possible enemy. After the right T cell is activated, your second line of defense awakes and rises like a teenager that needs to get up on a school day, very slowly. Your heavy weapons are incredibly effective, but they're not fast. The activated helper T cell begins to clone itself over and over again. 1 becomes 2, 2 become 4, until there are thousands of them. Now they split into 2 groups. The first group quickly moves to help out your soldiers. At the battlefield, things are getting out of hand. A tired macrophage is ready to give up. After fighting for days, it just wants to go to sleep, like many of its buddies have done already. But now the helper T cells arrive. One of them comes to the tired macrophage and whispers something using special chemical signals. In a heartbeat, the demoralized soldier feels fresh again. But there's something else. A hot, white anger. The macrophage knows what it needs to do. Kill. Invigorated, it throws itself against the enemies once again. All over the battlefield, this begins to happen. Meanwhile, the second group of helper T cells was working on activating another line of defense, B cells, your antibody factories. Antibodies are protein superweapons that look like tiny crabs with 2 pincers to grab enemies. Just like the helper T cells, there are B cells in your body that are able to make just the right antibodies for every possible enemy. And the helper T cell is looking for exactly these B cells. After a day or 2, the right B cell is found and begins to clone itself. As soon as enough clones have been made, each B cell begins pumping out up to 2,000 antibodies per second. About a week after you injured yourself and bacteria invaded, your second line of defense finally arrives in full force. The tiny army begins to saturate the battlefield, pinching and stunning desperate bacteria. The antibodies clump them together and make them unable to move or fight, while your soldiers massacre the defenseless victims. The tide is turning fast. As the last enemies are cleaned up, your soldiers realize they are no longer needed and begin to kill themselves to save resources. But not all of them. A few helper T cells remain and turn into memory cells. They will guard the tissue for years, making sure the same bacteria will never again gain a foothold here. Similarly, a few B cells will stay alive and keep producing a low amount of antibodies, making you immune against this bacteria, maybe for the rest of your life. One day you wake up and notice that the wound has grown over and left nothing but a faint red mark. You were completely unaware of the drama your cells had to deal with. For you, the whole ordeal was a slight annoyance, while for millions of cells, it was a desperate fight for life and death. But this is just the beginning of the epic story.
Yeah, so hopefully that video really sums everything up. This is the end of today's class. Feel free to leave. Next class, we'll be starting our new units, and it's all going to be about ecology, evolution, and biodiversity and things like that. So stay tuned. Yeah, have a good week.